Okay. Pause it until you start. Share. Pause recording. No, really. Okay. Okay, well, thanks for joining us today. It is my pleasure to introduce Coryna Hebert, who will be defending her master's thesis today. I want to share a few things from her past that prepared her for coming to graduate school and for this day. One of the things that I remember when we got this fateful email in around January 2019 asking about a graduate opportunity in the lab was Coryna has spent 13 months on an atoll in the middle of the ocean. I figured anybody who could handle living by themselves or with one or two other people working their tails off in the middle of the ocean, I want that person in my lab. Also, interestingly, some of her undergraduate research projects, I kind of forgotten about this, but she looked at feeding behavior of eastern gray squirrels and also did an independent project on mud tub formation of a marine polychaete. So she has a very diverse background, but also has a ton of work experience in wetlands and restoration, and particularly as a botanist, as a plant enthusiast. And when she emailed me in January 2019, I did not have a graduate position opening yet, but what we did need was an awesome field tech for the summer. And I knew that this was going to be her, hopefully her opening into the lab. So she worked closely with Ray Robinson that summer and was really integral in helping us launch a whole series of experiments that did spawn some interesting observations that really, I think, helped her figure out the focus of her master's work today on microtopography, as well as seed mixes and restoration. So Coryna, I'll turn it over to you and you can share what you found.
All right, thank you. Great, yeah, thanks for that introduction, Karin. Let me just minimize this. Okay, and thank you everyone for coming today to hear me talk about my master's research on the effect of species choice, seed mix composition, and microtopography on native plant restoration in Great Salt Lake wetlands. So to begin, I want to give everyone an overview of my talk today. In the introduction, I'm going to talk about our study system of Great Salt Lake wetlands and contextualize the need for restoration in the system. I also want to define microtopography and discuss why it's specifically important in wetlands. In my first chapter, we conducted an observational study to characterize microtopography in Great Salt Lake wetlands. And then in my second chapter, we conducted 4 experiments to test these several different restoration strategies, these being species choice, seed mix composition, and microtopography. So as many of you I'm sure are familiar, along the eastern shoreline of the Great Salt Lake, there is a vast system of wetlands. This is very significant because the Great Salt Lake wetlands comprise most of our state's wetland habitat. It's about 70%. These are a continentally significant resource for both migratory and resident bird populations. Unfortunately, the system is extensively invaded by a non-native grass, Phragmites australis. Phragmites grows in these really dense monotypic stands and spreads really aggressively, both vegetatively and through prolific seed heads. Wetland managers have found some good techniques to contain large stands of Phragmites. This typically includes a 3-year cycle of herbicide, mowing, and grazing, or some combination of those techniques. However, once large stands of Phragmites are contained, native plants don't generally return on their own, meaning that the seed banks that are left are generally insufficient to revegetate native plant communities. So that kind of brings this need for seed-based restoration. So given the scale needed in the system, because the Phragmites invasion is so extensive, seeding as opposed to strategies such as planting individual plugs of species is just a more logistically feasible strategy. So moving forward, when I talk about restoration in this system, I'm talking about ways to improve those seed-based restoration outcomes. However, in this system and wetlands in generally, there are a lot of challenges to restoration. So the first is that wetlands are naturally dynamic systems. Water levels fluctuate frequently, and sometimes water levels fluctuate pretty unpredictably. And this makes seed-based restoration pretty challenging because really subtle changes in soil moisture, light, and temperature, and just general environmental conditions can impact the germination and establishment of native plants from seed. Additionally, in our system, as I'm sure many of us are aware, Utah has experienced extreme drought conditions in recent years. And if you're trying to revegetate a wetland with native wetland species that typically like wet environments, having extreme drought conditions is going to pose a really big challenge to that. And then finally, even once Phragmites is contained, that doesn't mean that we're still not facing really high invasion pressure. For reference here, this is a Phragmites seed head, and one seed head contained thousands of viable Phragmites seeds. So even once large stands are contained, there's going to be a continual pressure of invasion from Phragmites. So I just want to sort of contextualize my broader research questions by chapter. So in my first chapter, we're just curious if there was microtopography in Great Salt Lake wetlands. And if so, if microtopography is associated with certain wetland types or wetland plant communities. And then in my second chapter, we looked at several restoration interventions to see which would improve seed-based restoration outcomes. These interventions were looking at species choice, seed mix composition, and microtopography. And before I get any further, now you're probably very curious what microtopography is. It's pretty much exactly as it sounds. It is simply a small change in the elevation of the soil surface at the scale of individual plants. Typically, this is less than a meter. We can think of microtopography as these two distinct metrics of relief and roughness. So relief is simply that change in elevation from a high to a low point. Roughness, we can think of as the path along the soil surface over a fixed distance. So like on the right-hand side of this image on the left, if we straighten out those squiggly lines, they would be longer relative to the ones on the left. And then in the real world, in a wetland, this is kind of what that looks like. This green line is representing the roughness and the blue arrow is the relief. So why is this important in wetlands? Well, there is a lot of evidence that microtopography is a really important structural feature of wetlands because it impacts soil moisture, hydrology, and biogeochemical processes at really small spatial scales. In lots of different wetland types, microtopography is typically associated with certain plants or certain species are known to form microtopography. An example of this would be Carex stricta, or generally a lot of Carex species that form these tussock meadows in the prairie pothole region of central North America. But there are lots of other examples. This is just kind of one that I think forms really obvious dramatic microtopography. Why is microtopography potentially useful in wetland restoration? Well, there's a lot of evidence in the literature that microtopography can improve restoration outcomes for several different reasons. In one context, microtopography increased native plant biodiversity. Microtopography can also buffer against hydrologic inconsistencies by increasing the water holding capacity of a wetland. And in another study, microtopography in a mitigation wetland was shown to improve the establishment of wetland species. But before we tested microtopography as a restoration strategy, we sort of just had these broad questions about microtopography in our study system, because to our knowledge, nothing had been formally recorded about microtopography in Great Salt Lake wetlands. So we were mostly just curious, is microtopography associated with certain wetland types or plant species around the Great Salt Lake? And I just want to kind of take an aside here and say that, some of our curiosity was also driven by these observations that things we were seeing in our study system. So this photo on the left, you see these plants kind of growing in these really distinct rows. And from, it looked like to us that this was happening because they drive these equipment called a marsh master through the wetland, and it makes these really distinct tracks everywhere. And it looked to us like that was really dictating where plants were growing or not growing. And then you see things like this, which is alkali bulrush, they're germinating in these little like cracks in the playa where there's more soil moisture. So it did appear that, these subtle variations in the elevation of the soil surface had large implications for, the spatial distribution of plants in the system. So I mentioned, we're curious about microtopography across wetland types. So I want to explain that, the Great Salt Lake wetlands are not one uniform type of wetland. There are several different types that vary in their hydrology and vegetation. So we have emergent marshes that are permanently to semi-permanently flooded, usually with deeper water. The dominant vegetation in this system includes cattails, bulrushes, also Phragmites tends to really thrive in these conditions. There are wet meadows, which are seasonally or shallowly flooded or saturated. Dominant vegetation in wet meadows includes saltgrass, herbaceous forbs, rushes, and sedges. And then finally, we have playas. Playas are temporarily and shallowly flooded and generally are always going to dry out at some point in the growing season. Common vegetation in playas include saltgrass and pickleweed, and typically playas are more sparsely vegetated than the other two wetland types. So we conducted plant and microtopography surveys in these line point intercept surveys across 14 sites along Great Salt Lake wetlands. So to do this at each site, we laid out 3 25-meter transects in a spoke pattern, and along each transect, we dropped a pin every 50 centimeters and recorded whatever plant species the pin hit. And then along the same transects, we also measured microtopography 5 times for a total of 15 measurements per site. We also looked at some additional site metrics, such as grazing status. To give you some context, grazing is a really common practice in Great Salt Lake wetlands. So, obviously that could impact microtopography. So it's an important thing to look at when doing the survey. Additional metrics also included the proportion of invasive species or native cover and litter cover. And I'm sure many of you are wondering how we measured microtopography. We use this very simple low-tech tool called a profilometer. As you can see, it's just a big metal frame. Inset into the frame are these 250 tiny pins. And then we can kind of pull a rod out from under that, and the pins mimic the path along the soil surface. We take a photo of it, and from that photo, we can glean those 2 metrics of relief and roughness that I talked about earlier. And this is kind of generally what we found. These are the median relief and roughness values by site. Roughness is there on the left, relief is on the right. And to orient you a little more to these graphs, they're colored by wetland type. So green are emergent wetlands, yellow are playa wetlands, and blue are wet meadows. It's kind of the general takeaway from this is that our emergent wetlands sort of had higher relief and roughness values than our other 2 wetland types. However, we're seeing that the microtopography in the system is overall pretty subtle. It's really only ranging from about 4 to, most of our sites have a relief of about 4 to 8 centimeters, which isn't really a huge change in the soil surface elevation. But to try to get at some of those associations between our sites and dominant plant species and microtopography, we conducted an ordination analysis, or an NMDS. So for those of you unfamiliar, ordination is just a really useful tool that helps you visualize the similarities or dissimilarities between different plant communities So if two of these dots, which are our plant communities by site, are really close together in this ordination space, it would mean they have pretty similar community compositions. So again, our sites are colored by wetland type. The green is the emergent, yellow is playa, blue is wet meadow. And then the additional benefit of doing this ordination analysis is we can look at these environmental variables and graph these as these vectors. So the length of the vector also indicates the strength of that environmental variable and its association with your plant communities. So just bear with me. I'll just go through some key things that we see in this ordination analysis. So the big takeaway for me was that emergent wetlands were more strongly associated with microtopography, especially this metric of median relief. Oh, and then, yeah, we also looked at, we can pull out some dominant plant species. So that's what those 4-letter codes are in this NMDS. And this association between microtopography and our emergent wetlands appears to also be driven by a couple of things. So we have Chenoplectus americanus, which is 3-square bulrush. Those sites that was the dominant vegetation component at those sites. I also want to note that those sites were not grazed. So it's possible that we're seeing more dramatic microtopography in emergent wetlands that had not been grazed by cattle before. So, I mean, ultimately, a lot of what we were observing was driven by a few sites that we went to that were dominated by Chenoplectus americanus. And I do think it's obvious from our observations at those sites that this bulrush species does form a pretty natural type of microtopography. This photo on the left, like they're growing in these big tufts, and between the tufts are these sort of eroded channels where there was a lot more water. But, you know, it would be really interesting to look at more ungrazed sites to see if there are other species in this system that form microtopography on their own. My sense of it is that grazing forms its own unique type of microtopography. I think that was some of the microtopography we were measuring at a lot of our sites, but it might obscure our ability to know what other species form natural microtopography because grazing is such a common practice in this system. So it would be pretty interesting to survey a lot more ungrazed sites, which again is kind of difficult because that's almost the entirety of the Great Salt Lake wetlands. But I mean, grazing is also a really important tool that managers use to control Phragmites. So it's not necessarily a bad thing or anything. It's just what we sort of observed across the study. So I'm going to switch gears here into my second chapter, where we looked at which restoration interventions will improve the establishment of native plant communities from seed. So these 3 interventions were species choice, which we looked at in a greenhouse experiment, seed mix composition, which we addressed in a greenhouse, mesocosm, and field experiment. And again, microtopography, since we were interested in also testing it as a restoration strategy, which we looked at in a mesocosm and 2 field experiments. So before going into the specifics of these 4 experiments, I want to take some time to discuss this concept of species choice. So broadly speaking, we had sort of 2 general groupings for all of the restoration species that we looked at across these experiments. The first one was perennial graminoids. Graminoid is just a fancy plant term for anything that resembles a grass, but also includes grasses. So in our species, this was bulrushes, a spike rush, and 2 grass species. Generally, perennial graminoids are going to be slower growing from seed. They are very narrow leaved. I think of like the blade of a grass. A lot of our restoration species to establish from seed and become a full-grown adult plant do require higher soil moisture. However, once these species are established, especially the bulrush species, they do provide a lot higher quality avian habitat. They are sort of the preferred species that managers want to see revegetated in this system. As well, a lot of the seeds of bulrush species provide really nutritious seeds for waterfowl. And then we have our forbs species contrasted to our perennial graminoids. Some of the differences between forbs and graminoids: forbs are generally, they're herbaceous flowering plants. The ones we looked at are either annuals or perennials. Generally, we have this hypothesis that they're going to be faster growing from seed. They are broader leaved, which could be a potential benefit in restoration because broad leaves mean they might be able to preempt other resources like light from an invader. And we also have this hypothesis that they're able to have a wider range of moisture tolerances, so they can grow in different soil moistures, unlike some of our perennial graminoids. And although they might not be, these optimal habitat species, they might have other ecosystem functions. Since a lot of them are flowering species, they might better support pollinators in our system. So these were all the perennial graminoids that we looked at across our 4 experiments. They were bulbous maritimus or alkali bulrush, Chenoplectus acutus or hardstem bulrush, Eleocharis clustris or common spikerush, Distichlis spicata or saltgrass, and Puccinelia nataliana or Nuttall's alkali grass. One thing I kind of want everyone to pay attention to here is the wetland indicator status of these species. So a wetland indicator status denotes the probability of a species to occur in a wetland. So if a species is an obligate species, that means it's basically going to occur in a wetland. You are not going to find it outside of a wetland. And so likely it has a lot of adaptations to growing in really flooded soil conditions or those hydric soils. And then so that's our 3, our bulrush and spikerush species are all obligates. And then our grass species are facultative or facultative wet, which, we're thinking means they might be able to grow in a wider range of soil moisture conditions. We have our forb species. We have Polygonum lapathifolium or pale smartweed, Rumex maritimus or goldendock, Lobium ciliatum or willow herb, Biden cernua, which is nodding beggartick, Symphyotrichum ciliatum, Raylus alkali aster, and Euthamia occidentalis or western golden top. So you can see here, most of these species are facultative wet, contrast to a lot of our graminoid species, which were obligate. So again, we have this hypothesis that perhaps they can establish across a wider range of soil moisture. But to test this, we first conducted a greenhouse experiment in early 2020, where we looked at the effects of water level, species choice, and seed mix composition on native plant performance. So we looked at 9 of these species individually and 2 different seed mix compositions. Our first seed mix contained only those perennial graminoid species, and the second seed mix contained perennial graminoids with fast-growing forbs. And we tested this across 3 water levels, a dry, moist, and saturated condition. We achieved these different water levels by filling up these plastic reservoirs with different high and low levels of water. So we checked the water levels once a day. And we collected percent cover data once a week. The experiment ran for 6 weeks total. Analyzed our data with a mixed effects. And this is generally what we found in this experiment. This is the final proportional cover by water.
You've heard everything.
3 different water levels. Saturated on the y-axis, we have our mean cover proportion, and this is by individual species. So the main takeaways here is that we were seeing that our forbs, as we had predicted, were more successful across all of our water levels. So those are those sort of reddish to yellowish species on the top. Our grasses are kind of right in the middle, Distichlis spicata and Puccinelia nataliana. They are more successful than our bulrush species. And unfortunately, our bulrush and spikerush really only establish in those saturated conditions, which is kind of what we had predicted. And not too surprising, but good to have this data to kind of back up our predictions about how these species were going to perform. And then this shows the cover by water level over time, over the length of our experiment. So in addition to just having higher cover at the end of the experiment, we can see that our forbs are growing faster than the perennial graminoids. You know, a lot of them are achieving close to 100% cover, like halfway through the experiment, when a lot of our graminoid species don't even have 50% cover. As well, our graminoid and forb mix really outperformed our mix that only contained perennial graminoid species. And so if you're curious about the breakdown of those individual species in our mixes, we have our graminoid and forb mix on the left across our 3 different water levels. And you know, the main takeaway here is that in this mix, the cover is almost entirely those forb species. The forbs like really outcompeted our perennial graminoids and comprise almost all of the cover across all of the different water levels. And then in our graminoid only mix, saltgrass appears to be kind of the winner among all of these species. You know, it establishes much more than our bulrush species. So we also wanted to look at how these seed mix compositions were going to perform in a field environment. And we also wanted to start to look at microtopography as a potential restoration intervention. So in our first field experiment in the summer of 2020, we looked at the effect of seed mix composition and microtopography on native plant establishment. And so we were curious if artificially induced microtopography would improve the establishment of some of these native species from seed. So we tested 3 distinct microtopography treatments and looked at the same 2 seed mixes, so the perennial only and the the graminoid only and the graminoid and forb seed mix. This experiment was conducted at Farmington Bay Wildlife Management Area, and we collected plant cover data again, which we analyzed with mixed effects models. As I mentioned, we had 3 microtopography treatments. These were high mounds that had a 15 centimeter relief, low mounds that had a 5 centimeter relief, which is kind of what you can see here. I think those are the high mounds actually. The idea with the mounds was that, you know, based on some of those observations we saw where little changes like having elevated soil surface was kind of allowing some forbs to grow really early in the growing season, we thought it might provide germination microsites for some of our forb species. And then our third microtopography treatment were ruts, which were dug 10 centimeters below the soil surface. The idea with the ruts was that, you know, perhaps if our site dried out, they would maintain some soil moisture further into the growing season. And the fourth was an untreated control. So this is what we found from this experiment. We also had the opportunity to go back in 2021 and collect the same cover data. So this is what you're seeing is the comparison between 2020 and 2021. The top panel of graphs is the difference between summed invasive and native cover by our seed mix treatment. So it goes control, graminoid, and then graminoid and forb. And the bottom panel of graphs is the individual species breakdown of cover. You are not seeing microtopography represented in any of these graphs because we did not find that it had an effect. So the main points here, so in 2020, we found that seed mix had an effect on native cover. So seed mix increased native cover relative to our control, but there was no difference between our seed mix type, which was pretty different from what we were finding in the greenhouse experiment where our graminoid and forb mixes had a lot more cover than the graminoid only mix. As well, although seeding increased native cover, it didn't suppress invasive cover, if that makes any sense. So there's about the same number or about the same cover of invasive species in our control and seeded plots. And this kind of contextualizes our second season results a lot, where we're seeing really high invasive cover and much lower native cover. So that effect of seeding that we observed in the first season didn't persist into 2021. And in this bottom graph, I'll mostly just point out that the yellowish panel is Phragmites. So they kind of just took over all of our restoration plots, which was a little disconcerting, but you know, this is what happens in the field. It's really hard to replicate some of those things we see in controlled environments in a field setting. So we wanted to do another controlled experiment, again, to look at microtopography in a more controlled environment and to look at how our seed mixes would perform with some Phragmites competition. So in 2021, we looked at the effect of microtopography, seed mix composition, and water level on native plant establishment and invader suppression. And we did this in these small mesocosms, which were outside of the research greenhouses. Mesocosm in this case is a fancy word for a children's kiddie pool. And in this experiment, we tested 3 types of microtopography. So 2 were just these system of ruts. And the reason we wanted to test ruts is because it parallels another field experiment that I will talk about after this. And it's a lot more tractable to reproduce at scale. And we looked at these across 2 different water levels, a high and a low. And again, we looked at the same 2 seed mixes, a graminoid only mix and a graminoid and forb mix. And we actually sowed our seeds at a density that was 3 times higher than what we used in the field experiment because our density seemed insufficient in the field. And then as I mentioned, we also added Phragmites competition. And we, at the end of the experiment, harvested all of our biomass, and that's what we used to do our data analysis with mixed effects models. Okay, so here we are seeing native cover on the left, Phragmites cover on the left, or sorry, on the right, between our 2 seed mixes. And again, I'll just mention, you're not seeing microtopography represented in any of these results because we did not observe an effect of microtopography. So yeah, this is on the left is average native % cover over time. The green is our graminoid and forb mix, and the orangish yellow color is the graminoid only mix. So basically what we're seeing is the graminoid and forb mix, similar to the greenhouse experiment, had higher native cover throughout the length of the experiment. And sort of the opposite was true with Phragmites cover, whereas the graminoid only mix had higher Phragmites cover throughout the length of the experiment, meaning the graminoid and forb mix was a little more effective at suppressing Phragmites competition. And this is the individual breakdown of biomass to kind of contextualize these graphs a little better. This is what our kiddie pools look like at the end of the experiment. So the graminoid only seed mix is on the left, and then the graminoid and forb seed mix on the right. So it's pretty obvious which one kind of is the winner in this context. And we actually had intended to run the experiment a bit longer, but our forb species were just so giant and we're starting to shade our other treatment that we had to harvest the biomass a little bit earlier. So on the left here is the average biomass in grams by species of our graminoid only mix. And again, pretty consistent with what we observed in the greenhouse experiment. Most of the biomass is saltgrass. Some of the other species, they still established, especially alkali bulrush or bulboshonis maritimus in the higher water level. But nothing is really coming quite close to our saltgrass biomass. And then for the graminoid and forb mixture, almost all of the biomass is represented by these 2 species, Biden cernua and Rumex maritimus, which are 2 of our forb species. And it seemed like they were pretty effective at suppressing everything else in the seed mix. It's really all we observed across all of our treatments. So finally, Although we didn't observe an effect of microtopography in our controlled environments, we did still want to look at it as a restoration intervention for a larger scale seeding effort. So in this experiment, yeah, we just looked at the effect of microtopography on native plant establishment, and we collaborated with the Utah Division of Forestry, Fire, State Lands and the Utah Division of Wildlife on one of their larger scale seeding efforts. So the type of artificial microtopography we tested was made with this piece of equipment called a disc roller that is sometimes used in mitigation wetlands to create artificial microtopography. And I think when we were experimenting with it, it did seem like there could be some potential benefits to using it. One might be that it might help hold seeds in place, which sounds really simple, but sometimes in a wetland restoration, if you're throwing a bunch of seeds on the ground and you have a flooded site condition and all of your seeds wash away, you can't really monitor the success of your seeding effort. It was also in drier areas, could potentially increase soil moisture and have little germination microsites that are holding moisture further into the growing season. Also, when we were kind of experimenting with this method, it was helping to break up thin layers of Phragmites litter, which is really beneficial because that can improve the seed to soil contact. And so, as I mentioned, this was a collaboration with wetland land managers, and we seeded 500 pounds of alkali bulrush, hardstem bulrush, and common spikerush at this bulk rate of 47 pounds per acre across the teal unit at Farmington Bay Wildlife Management Area in May of 2021. If anyone is curious what 500 pounds of seeds look like, that's that right photo. It's probably more like really 150 pounds. It's just a portion of the seeds. It's a lot. So our method here was we used a broadcast seeder attached to this equipment called a Marsh Master. I think I mentioned earlier, it's basically like this big aluminum tank that they drive through the wetlands. So we drove the seeder adjacent to the disc track so that we could have some seeds in both the disc areas and non-disc areas for comparison. And we went back twice to monitor. We established 50 paired 1 meter squared quadrats. So one quadrat was in the disc tract, the other was in the non-disc tract. We came back in the middle of the growing season in July and once at the end of the growing season in September. And I'll just point out, this is some of what we were observing at this mid-July point. This wasn't what all of the site looked like, but we did get kind of excited when we saw this, because we saw a lot of alkali bulrush germination in the disc tracts. Like you can kind of see they're growing in these line formations. But you also might notice that the site looks really dry, which is, something that's hard to predict what the water levels are going to be like when you do these large scale seedings. So we weren't sure if, any of our species were necessarily going to survive until the end of the growing season. And that's basically what had happened. So this is the alkali bulrush cover between our disc and non-disc tracts at those two monitoring dates. I'm only representing alkali bulrush because we did not observe really any of the other two species or trace values occasionally. So at this first monitoring date on the left, we did see higher alkali bulrush cover in our disc tracts compared to our non-disc tracts. But obviously it's not a huge difference, like 1.5 to 3%, but they were seedlings. So had they survived, it might have been a larger difference later in the growing season. However, we observed this really high mortality between monitoring dates, probably due to the very extreme dry conditions at our sites. Because if you remember, alkali bulrush is an obligate wetland species, so it does need some water to survive. So, I think the results of this monitoring are pretty ambiguous, but, based on some of our observations, I think there might be some benefits to disking, but I can't really say that, it's the best method given what we saw. But however, disking is pretty, it's inexpensive. They already have the equipment available. I think it's worth continuing to look into as a potential restoration intervention. So to just kind of give you some summary across our experiments, we saw that native plant performance was pretty dependent on experimental environment. Our seed mixes performed sort of as we had predicted when we had these controlled environments, but it's really hard to replicate those results in a field setting, likely because of those things I was talking about earlier regarding sort of the general environmental uncertainty of working in the field, specifically really high invasion pressure from Phragmites and unpredictable water levels. As a restoration intervention, microtopography was mostly ineffective, but again, disking may have some benefits. I would say that our winning species across all of our experiments would include Distichlis spicata or saltgrass, Biden cernua, nodding beggartick, Rumex maritimus, goldendock. And although none of our winning species were our bulrushes that we say provide, the highest quality avian habitat, finding any species that can outcompete Phragmites, especially at those early life stage cycles, it is a really valuable contribution to restoration in the system. Distichlis spicata, also there's evidence that it does really well after an ecosystem disturbance, like it's often an early colonizer, which is probably why it is such a successful restoration species. And as I mentioned earlier, there might be other ecosystem value to using these other forb species that we just haven't quite identified yet, such as improving pollinator habitat. So and then some general summary and conclusions. I think based on our results, widening species choice to incorporate fast growing forbs was the most promising restoration strategy to achieve higher native plant establishment. But I do think on its own, it's not going to be enough to establish long-term native plant communities. I think that's going to require a really multifaceted approach across longer temporal scale, such as multiple years of seeding with higher sowing densities and just a really, vigilant control of invasive species at a restoration site. But I do think that incorporating fast growing forbs can sort of help tip those scales towards native plant communities. And that is all that I have today. I would like to thank everyone that helped me along the way in my research, especially my advisor, Karin, my committee, Kari and Joe, for all their incredible feedback along the way, all the members of the Wetland Ecology and Restoration Lab and the land managers on the Great Salt Lake, and of course, all of my amazing family and friends for their endless love and support. And with that, I'm happy to answer any questions.
Check your comment box there. Well, people are thinking of
Oh, okay,
questions.
thanks.
You've got one on there, I think. Any questions for Coryna?
Freda.
So I'm curious because I'm clueless about seeds and plants in general. When one of the things that you tried was increasing the seeding density,
Mhm.
how practical is that on a large scale in terms of like cost to wetland managers and finding the seeds and all that kind of stuff?
That's a really good question. I think that is a challenge for wetland land managers, right? Because if they have a fixed amount of seed, they want to spread it over the largest area possible. But it might just have to be like a cost trade-off thing where, if you're going to spread your seed out so far that none of the seeding is successful, maybe you just have to target a smaller area to revegetate native plant communities. As well, the forb species we were testing aren't generally commercially available, or at least not yet, but I think they could be if there was a demand for them. But yeah, so the experiment where we did collaborate with land managers, we tried to use a higher seeding density that was maybe around like 2 times through that base density. So it's not, it's something they can do, but I think for them, it's, you know, they're going to have to accept that maybe they're going to have to look at a smaller area.
We do have a question from Peter Wilcock here.
Oh, okay.
Please say more about the soil surface, vegetable versus mineral. It looks like your field measurements represent a vegetative surface, whereas your experiments tested variation in mineral surface elevation. So what were you measuring when you were using your profilometer?
Oh,
Vegetable
yeah,
versus
that's a
mineral.
good question. Thank you for, okay. Yeah, so when we use the profilometer, we are specifically measuring the soil surface. And sometimes that meant moving litter out of the way and like adjusting each pin to ensure that it wasn't hitting any kind of like decomposing litter, because we really wanted to get those soil surface elevation measurements. Although, you know, I mean, it's kind of one of those things. It's like, well, what is the soil? At what point is decomposing litter part of the soil? And at what point is it not the soil, you know? But yeah, we generally tried to move all vegetation material out of the way to get those measurements.
So if you hit a base of a plant, then you would move the profilometer over then?
Well, the advantage to the pins is like you can go kind of through a plant. So if you have like a big tuft of, you know, say 3 square bulrush or something, we could just kind of, the pins could kind of go in between it and still kind of get that wherever it's like accumulating soil underneath the plant. Katie.
In your first chapter, did you only measure microtopography at each of your sites once, or did you do it over time?
Just once.
And did you, I'm sorry, I'm asking these questions to refresh my memory. Did you, it was your wettest sites that had the most microtopography differences?
Yeah.
Do you expect that microtopography in these dynamic systems is dynamic based on like differences in water level and saturation, like moving sediment, or would you expect if you went back maybe in the same season that it would be similar, grazing aside, like not cows stuff?
I think it's probably, you know, dependent on how established maybe the vegetation is at that site. Like 3 square bulrush is a perennial species, so even if it doesn't come back every year, it's gonna still like have all the same root structure. So I would expect microtopography maybe in those sites to be a little less dynamic. But, you know, we've seen a lot of changes in plant community structure at some sites from like one year to another. So when that's the case, like in some of these wet meadow sites that have maybe more annual species, I would expect microtopography maybe to be more dynamic across seasons.
We do have another question on there from Molly Blakowski, who would like to know about, can you discuss the decline of Great Salt Lake and how that may impact invasive species management strategies in the wetlands?
Oh, sure. I mean, yeah, I mean, as we all know, the Great Salt Lake has been at like a historical level for the past couple of years, or was recorded at the lowest level ever in 2021. And that's, you know, definitely going to affect wetland management and has, I think, contributed as well to the expansion of Phragmites into the lake bed. So I don't really know how that changes, you know, invasive plant management for some of the wetland land managers. I think it is really challenging. Yeah, how's it going to impact invasive species management strategies? Yeah, I think it just means that there's probably more Phragmites, but then, you know, is it always the best strategy to remove Phragmites immediately as it's like receding into the lake bed? I think that's really hard to say because, you know, Phragmites is also like stabilizing those soils. So yeah, I don't know.
Another one, you might have said from Spencer Good, you might have said this, but in your seeding experiments, did your seeding rates vary by species? would there have been possible competition within the mixes that influenced the overall mix performance?
Yes, they did vary by species. So for most of our seed mix densities, it was a 1-to-1 proportion of species. So, if you have a seed mix with 5 species, it would be 1/5 of the seed mix. With 9, it would be 1/9. But we reduced the portion of forbs in our field mix because of what we observed in the greenhouse experiment. So the forbs in that context were 25% of our seed mix. And can you repeat the second part of the question again?
Would there have been possible competition within the mixes that influenced the overall mix performance?
Oh yeah, definitely. And I think we observed that, especially in our controlled environments where we didn't see anything else that we had sown, especially in the mesocosm experiment. It was pretty much dominated by 2 species. So, that's a thought as well, is like, well, is it best to sow these species together then if they're just going to outcompete one another? Or should you just maybe use like a really high density of these species that perform really well and then maybe try to seed other species later on? Joe, you had a question?
Yeah, I was just gonna, sort of related to Peter's, but why'd you use such a simple, silly tool instead of something fancier like ground-based LiDAR?
Yeah.
There's a lot of nice instruments out there.
This is where you
That's
can
true.
say, Joe, this is because you told me. to."
Yeah. Firstly, because you told me, but you expanded on, why these remote sensing methods, although, they're amazing and very fancy and high tech. We have a lot of really dense vegetation in our system, and those remote sensing methods can be pretty easily skewed by that vegetation, or even really dense litter mats. So you can't really be certain that you're actually measuring the ground, when that was sort of the advantage with the profilometer.
It's related to that. Do you feel like it did a good job of capturing microtopography?
I do, yeah. I mean, when you're out there like physically adjusting the pins, I do feel like we were capturing that, path along the soil surface. I will say there were some issues using it in some emergent sites. And one of the reasons we have a few less emergent sites was because when I went back to measure microtopography in, say, like a cattail stand, the sediment is so mucky that, it was like, oh, I don't actually know what I would say the ground is, so some of those 3 square bulrush sites, the soil was really dense clay. And so the pins kind of stop even if the site is inundated with water. But yeah, and using the profilometer in some wetland contexts is tricky. And I don't know what method you would use to measure the soil surface in that context. Marshall.
Yeah, so I'm curious, a lot of your kind of observations and then field scale topography data kind of illustrated that maybe topography is important, but then when you got it down to the small plot scale,
Mm-hmm.
where the microcosms and the greenhouses, it
Yeah.
wasn't really factoring. And I'm
Yeah.
curious, like, what you think might be controlling that. Is that like soil structure or hydrologic influence, or like how much influence your topographic changes are having?
Yeah. So I think the fact that we didn't observe an effect of microtopography, the reasons were maybe different in either environment. I think in the first controlled field experiment, there is just so many invasive species that we observed this effect of seeding, but and it's also such a small area. I think in that case, microtopography just wasn't important. It couldn't overcome the amount of invasive species coming up from the seed bank. And I think in the mesocosm experiment, the idea between the high and the low water levels was that at the low water level, the ruts were supposed to have more soil moisture, and the no microtopography treatment was supposed to be kind of dried and moist at the soil surface. But I think I just put too much water in the low treatments to where the microtopography didn't have the effect that I thought it would. And you can kind of see from like the biomass data, you know, water level did have an effect, but it was kind of small. Like there's not a huge difference in the low and the high biomass. And I really should have like drought stressed that lower treatment. And maybe then we would have been observed effects of microtopography, but that's kind of my sense of why it didn't work in that experiment.
Okay, well, let's thank Coryna for her presentation.
Thank you.
Okay, thank you so much. I feel like I knew a lot of it, from, like dinner conversation, but this was more informative. That's